Well, thank you, Christina. I appreciate you giving us this opportunity. I've recently had several people ask for me to put together a presentation to explain the PLFA results and the process. And this has worked out really well, and I appreciate you putting it together. So just to let you know what I'm going to be talking about, I'll talk a little bit about our lab background, our soil health definition, why we chose PLFA, the experience we've had with it, what PLFAs are, and then sort of the interaction with PLFAs, microbes, and their role in the soil. I will go through an actual soil health PLFA results report, and then just give you a little bit more information about PLFA and how we do it, and then at the end have time for questions. So while we're going through this, write down any questions you may have, and maybe they'll be answered, maybe they won't, and then we can discuss those in the end. So I am the soil health specialist at the Soil Health Assessment Center at the University of Missouri. We used to be a soil characterization laboratory. We helped the National Laboratory in Lincoln, Nebraska with NRCS with the Accelerated Soil Mapping Program in the mid-'80s. And then in 2012, we decided to add soil health analyses and have added space, people, new analyses as we've gone on. So just to touch a little bit about our soil health definition, we are mainly concerned with health being a condition where you have vital functions that are performed normally or properly. So in soil, that would include producing biomass. This is the ones people usually come up with first. And I've highlighted the ones that I believe that soil microbes are included in, just so you get an idea of the role of soil microbes in soil health. So you have rhizobia, fixing nitrogen, you have Arbuscular mycorrhiza that help the plants produce more food and fiber. So I believe they have a role in that. They help you regulate water flow and storage. They help in soil aggregation, which allows water to come in. The mycorrhiza help water get to the plants. Another soil function is filtering, buffering, and transforming matter. So I see the filtering part as being more a physical part, buffering is more chemical, but transforming the matter is what I see the main function of soil microbes being. Breaking down additives, herbicides, other organic substances that are added to the soil. And just in themselves, they store minerals and they're part of organic matter, and obviously they're part of the microbes or organisms in the soil. Supporting plants, I would say that mycorrhiza do that. I wouldn't say they help support structures all that much. They're not part of the raw materials. But I do believe that past climates, vegetation, ecology, civilizations, all that affects the soil long term and may also affect the microbes long term. I don't know exactly to what extent or how long, but I think a lot of these things will have an effect on the microbial populations on into the future after they have been changed. So when we started the lab, we were trying to get physical analyses, chemical analyses, and biological. And one of the biggest things that we had to figure out was what method are we going to use for analyzing soil microbiology? We had some analyses like active carbon that touches on the microbial population, but we were looking for something that gave us a little bit more information about the soil microbes. So there were sort of three groups we were looking at. There's some that are culture-based, some that are DNA-based, and then phospholipid fatty acid analysis. So the problem with culture is that a lot of the soil bacteria do not grow on standard media, and one of the culture methods we were looking at, it had a lot of different substrates, and I'm sure it gave a lot of information, but we still felt that this was not going to get a lot of the microbes that were in the soil, so that narrowed it down to DNA and PLFA, and this was 2012. DNA soil microbial analysis has advanced a lot since then. But at that time, it was more qualitative than quantitative. It did allow some species, the species that we actually know, to be identified, but a lot of soil microbes, we don't know what they are. We haven't identified them because we couldn't raise them on standard media. So in some ways, it's more specific than PLFA. Another thing is that DNA does not break down quickly in soil, so there's a legacy effect. So you don't know if those organisms are there today or last month or even last year. So with PLFA, it's quantitative. The total PLFA is very highly correlated to microbial biomass measurements. And this is also the living biomass because phospholipid fatty acids degrade rather quickly. So you're getting a snapshot in time of the microbes that are presumably living. And because of this, you can see rapid changes in the environment. And though it does not allow you to identify down to species, even for those we know, it can help identify groups of microbes like different kinds of bacteria, fungi, Arbuscular mycorrhiza. And since you can get a whole list of the phospholipid fatty acids that are available in a soil and how much is each one, it can provide a fingerprint for a particular soil at a particular time. So that's what we chose. So after we selected that as our primary microbial analysis, we purchased the equipment, software, and training in early 2013 from MIDI Incorporated. They have the Sherlock Microbial Identification System, and that is the software we use with our gas chromatograph. And there were different methods. They had just come out with the high throughput method. It was by Jeffrey Beyer, who worked at the Beltsville USDA Agricultural Research Service, and he worked closely with MIDI on a high throughput method. So instead of the extraction taking 4 days, it took just 2. So we went with that method. And that's what we've been doing ever since. We're primarily a service lab. We mostly do the analyses for other people. Sometimes we work with people who have grants and do the research here. We work with them, but a lot of times I don't know where these samples came from, and that's probably a good thing because people submit the samples, we run them, so we're not biased. I'm not sure if that one's right, maybe you should run it again or anything like that. So we primarily run research samples. We generally discourage farmer samples because as we go through this, I think you'll see that there's just so many variables that it's really hard to interpret just a single sample. And it's a pretty expensive sample or analysis for say, a farmer. But you do get a lot of data from it. So for a researcher, you're getting a lot of information. We've run about 15,000 samples from about 30 different states, about 3 different countries, and we've run analyses on samples for nationwide studies, a couple of them with NRCS, one with the Soil Health Division and another with the Soil Science Division. So we've also looked at it in soil characterization. How do the microbes change as you go down through the different horizons of the soil? So what are phospholipid fatty acids anyway? So when I found this, I liked it because I remember when I was a freshman in college studying for a biology exam, learning about what triglycerides are. They're glycerol and 3 fatty acids. So this is glycerol. It's 3 carbon chain with 3 OH groups. And if you add a fatty acid to this, that's a triglyceride. So these are 3 fatty acids. You'll notice that the fatty acids are different. The lowest one has a double bond. They can be different lengths. They can have branches to them. So now if you'll take the triglyceride and exchange one of the fatty acids for a phosphate group, this is a phospholipid fatty acid. So what do they have to do with microbes? So the phosphate group will form a polar head, oops, excuse me, and then the fatty acid chains are nonpolar. So if you remember from your biology class, phospholipid bilayer is present in the cell membranes of soil microbes. This is where they come in. So you can see if you can extract the phospholipid fatty acids from the soil, the more microbes, the more phospholipid fatty acids you can get. So you can see how that would be very highly correlated with the microbial biomass. Another thing is, again, you'll notice that these phospholipid fatty acids are different. And there are different kinds, or some fatty acids that are found mostly in one or another kind of microbe. So you can get some idea of what the community structure is, what general kinds and how many there are within the soil. So now I'm going to go through some of the major kinds of soil microbes that we can use to put in groups with the phospholipid fatty acid information that we get. Bacteria can generally be grouped into gram-positive and gram-negative bacteria. And what this is referring to is there's a person named Graham who came up with a staining mechanism so he could separate the microbes into two groups. You'll remember before you had DNA, you had to look at the shape of the bacteria, whether they were circular rods or spirochetes. You could look at what kind of colonies they were in, and this was just one more way to separate them out and identifying them. So a gram-positive stain was if you had crystal violet and it stained them purple. If you have gram-negative, it was not stained by crystal violet, so they counterstained with safranin, which made them pink. So this way they could separate the bacteria out into two groups. So the reason that this stained is, look on the bottom here, the gram-positive soil cell envelope has a thick layer of peptidoglycans, which would take in the crystal violet, whereas the gram-negative bacteria had two layers of these phospholipid bilayers. They had one on the outside and then a thinner layer of peptidoglycans and then another phospholipid bilayer. So these were the two groups of bacteria. So let's take it into the soil and look at how we get cued into which ones they are as far as phospholipid fatty acid analysis. So the gram-positive bacteria, they tend to have branched phospholipid fatty acids. So if you look on the left side on your screen, you'll see that there's a methyl group that's attached to one of the carbons, and the gram-positive tend to have it either on the first or second from the end. They'll have a branch of another carbohydrates. So in the soil, the gram-positives tend to be larger than the gram-negative bacteria. They tend to be widely dispersed in the soil. They decompose complex organic material that fungi and gram-negative bacteria partially composed. They're not as closely tied to the rooting zone as gram-negative bacteria are. They're resistant to water stress because they can form stress-resistant structures called endospores. So gram-negatives, they tend to have phospholipid fatty acids or fatty acids with that have a double bond, one double bond, or monounsaturated fatty acids. Or instead of having a double bond, they may have a 3-carbon cycle in the middle of it. And there's some evidence that when microbes are stressed, that they form these cyclic bonds, the 3-carbon cycles. But there may be organisms that just in general have the 3-carbon. So this is another thing you can look at. I'll talk about it a little bit later, but it's one of those that you may need to take with a grain of salt. So gram-negatives are smaller than gram-positive bacteria. They tend to be involved in all phases of the nitrogen cycle, and Rhizobium is a gram-negative bacteria. They predominate in the rooting zone, and they break down newly added organic matter, but they decrease in proportion as you go down the soil profile. They are more sensitive to water stress than gram-positive bacteria because they can't form endospores. Actinobacteria, and when I took soil microbiology, they were called actinomycetes, but they've since learned that these are actually gram-positive bacteria. But they have branches like gram-positive bacteria, but it tends to be on the 10th carbon of the chain. So they have a methyl group there, just a 1 carbon, 3 hydrogens. So similarities to gram-positive bacteria, but they have some differences. They're filamentous, which is why they were thought to be fungi. They reproduce by spores and fragmentation, similar to fungi. They produce geosmin, which is the smell of soil. So when you smell soil, when you're working up your garden, or when you go by a farm, if they're working up the soil, you smell that. That's produced by the actinobacteria. They decompose cellulose and chitin. So they're getting close to being able to digest some of the things that fungi can. And we've seen some evidence that in the absence of fungi, that actinobacteria might take up some of their niche or some of their role in trying to digest some of these more difficult to digest organic materials. They continue the decomposition of the organic matter that started by the saprophytic fungi. They tend to be drought and temperature resistant. They, like the gram-positives, which they are a part of, grow deep into the soil, but they're sensitive to acid soils, low oxygen, and wet or saturated conditions. These microbes produce special chemicals when stressed, and some of these are pathogenic. They participate in chemical warfare against other soil organisms, so that has made them useful to humans because we have gotten antibiotics from them producing these chemicals. So streptomycin, aureomycin have been discovered and produced commercially from the actinobacteria. So the next group is Arbuscular mycorrhizal fungi, and these are indicated generally by one phospholipid fatty acid in particular. And I'm going to take this time to show you, this is how the phospholipid fatty acids will be reported on your reports. The first number tells you how many carbons, and after the colon, you have 1. That tells you that there's 1 double bond. Then you'll have either a W or an omega and a number, and that will tell you what carbon that the double bond is positioned at. And these are cis. There are cis and trans fatty acids. Our analysis won't be able to tell them apart. You have to run a sample for about an hour, which you can imagine would take forever for us to run these analyses. If we took it that long, we run it for about 15 minutes. So this phospholipid fatty acid is mainly found in Arbuscular mycorrhiza. It can also be found in gram-negative bacteria. As we've learned, gram-negative bacteria are found with monounsaturated phospholipid fatty acids. So the mycorrhiza grow in long, thin hyphae, and they form mutually beneficial relationships with most plants. They colonize the roots and produce structures called arbuscles inside the plant cells. They provide the plants with drought resistance because, as you can see in this picture, they drastically expand the soil rooting, effectively expanding the soil rooting system, and allow the plants to take in more water and more phosphorus, nitrogen, sulfur, zinc, copper. And what the plants do in exchange is provide the fungi with a supply of carbon. Most fungi, saprophytic fungi, get their energy from breaking down carbon compounds, organic matter in the soil. Mycorrhiza are dependent upon the plants to provide the supply of carbon. They also protect the plant roots from disease by producing antibiotics and immobilizing heavy metals. Another thing they do is they release proteins through the hyphae to glue the soil particles together and promote soil aggregation. Soil fungi, the Actinobacteria and the saprophytic fungi, we use one phospholipid fatty acid to indicate these, and it's 18 carbons long. It has 2 double bonds at the 6th and 9th position. So if you've heard of omega-3 fatty acids, that would mean that they have probably 1 double bond at the 3rd carbon. So you've probably heard of 6 and 9 fatty acids too. So that's something to tie it into something that you've heard about. So the role of soil fungi, first off, it's a broad category. You have everything from single yeast cells to some of the largest organisms in the world. And I know in Michigan you have some of these fungi that take up acres or maybe square miles of territory. Some soil fungi cause plant diseases. Others live cooperatively with plants. Saprophytic fungi break down organic residues. They produce humus, release plant available nutrients. As they break these things down, they decompose everything from simple sugars to starches to resistant cellulose and lignin substances. They usually prefer more acidic soils than do bacteria or Actinobacteria. And because they're aerobic, they tend to prefer the soil surface horizons instead of going very deep into the soil. Another group that we look at are eukaryotes. So these are organisms that have more complex cell structure than the prokaryotes like the bacteria. Their cells contain membrane-bound nuclei and other membrane-bound organelles. They feed on soil organic matter, soil bacteria, plants, and each other, releasing and recycling the plant available nutrients. There are smaller eukaryotes, sort of like protozoans, amoebas, paramecium, and then there are multicellular ones like nematodes, insects, earthworms, acari, which are ticks and mites, and crustaceans. So these organisms are sort of the top of the food chain. The larger eukaryotes can mix the soil and create channels for water and air movement in the soil. And currently we don't list just protozoans, but if you wanted to figure out on their own, these would be they're the 20 carbon length compounds or fatty acids that are listed here. And then other eukaryotes are, excuse me, other polyunsaturated fatty acids, which means that they have more than just one double bond. I thought I would share a little bit of how it looks when we're running the analysis after we've gone through the extraction and separated out just the phospholipid fatty acids, and now we're putting on the gas chromatograph. This screen is what it looks like when we're running it. So this first part here on the left, that was actually a calibration standard. I can tell because it's evenly spaced peaks, and there's a tall one and the short one. That's what every 10 samples, it'll run a calibration standard just to make sure that it's running okay. And then this next one is just the beginning of running an actual sample from the extract of the soil. So what a gas chromatograph does is it has a column, and it doesn't look at all what you think of as a column. Missouri is famous for its columns. This is more along the lines of your intestine. It looks like a real thin little wire in a big coil. So the purpose of the column is to separate out the phospholipid fatty acids. So the short chain, the small phospholipids, will zip through there much more quickly than the long branched ones. So through 15 minutes, they get separated out. And then it's released to a flame ionization detector, and that's what will cause the peak to be made. So the amount of each one is the area underneath the peak. So that's, you have them separated, and then you have the amounts of each. And this is what it looks like, and you can just show for one sample, all the different phospholipid fatty acids that were found. You can show the chromatograph. What I tend to send people, because most of our people are doing research, is the results of a lot of different samples in a spreadsheet form. I actually download it from Access, from our software, and then export it into Excel. So now I'm going to go over the results reports that I send people. So when you get it, there are 7 pages. Your samples will be listed here, and we do list the state and the county, and actually there's a usually a code for who the, oops, let me go back, who sent it in, and then right in the middle is the designation for the samples that the clients have sent and the date that the samples were taken. The different pages that you get, there's one that's WT5_WT. That is talking about the weights, so it lists the phospholipid fatty acids in the sample by picomole per gram of soil. And for each one, so it lists all the different phospholipids that were found and how much was found in each one. And at the very right of it, as you'll see, I give the total and the number of phospholipid fatty acids. The next one, again, it lists all the phospholipids that were in the sample, but instead of in molarities, it's given as percent of the total phospholipid fatty acids. So this is relative abundance. So then we have 2. The first one is, it lists the microbial types into which the PLFAs have been categorized and summed up as units of picomoles per gram. And then the following page is, again, the microbial types, but listed or values given as a percent of the total that have been categorized into the microbial groups. Then we have 2 pages that are listed as fat types, like saturated, unsaturated, polyunsaturated, and then some of the different branched, straight chain, different types of fats. And one page will have it in picomoles, another will have it as the percent of the total categorized into the fat categories. And then the last page, there are 6 calculated ratios, and I'll go through each of these pages at a time here. And I have this small data set that I usually send out. It was sent in by somebody who works for the Missouri Department of Conservation. They have used these sample sets in training. It's from a farm that had been in CRP for a long time and then was going to be taken out. So we have that data. These are actual data that we'll be looking at. So this is to look at it a little bit more in depth, the WT5_WT file. So the individual PLFAs have all been listed here in picomoles per gram for each one of them. This is helpful to the clients because while we're using classifications for the different groups, for the different microbial groups, there are different people who do it using different methods. And so if you wanted to, you could sum up your own totals. And we have had clients who have done that. Probably the most used piece of data that is provided by this is the total PLFA, because this is strongly correlated with microbial biomass. So it's used to report microbial biomass. I also give the number of PLFAs. That just says how many different ones were in the sample. That is rarely used. It could sort of give information about the variability, but we don't really know whether there are a few organisms with a lot of different phospholipid fatty acids, or if there are a lot of different microbes with just a few fatty acids. So that's just to give you an idea of how many were in the different ones. Generally, it's not used for publication, but I just think it's a useful thing just to see. So this is just a screenshot, but otherwise you could scroll through and see all of the different phospholipids that were found. Just to look these samples over, you'll notice that probably the fourth one down and then the last one, just looking at those, they have the most microbes. I would say that they're probably the most healthy because they have the more microbes. The fifth one has the lowest. And then there's the one that's third, it's sort of medium, and then the top two are somewhere between the fifth and these others that I've said were the more healthy ones. So the next page is, it's giving you all the different phospholipid fatty acids as a percentage of the total. I would say this is probably the most seldom used of the information, but it can be useful. Recently, we have a client who is sending in samples from FENS, and they were wanting to know about heavy metals and whether there are sulfur-reducing bacteria. So there are some phospholipid fatty acids that are found in bacteria that are reduced sulfur. So I had looked at the first page and, oh, these have some really high levels of it. So I was thinking, well, maybe they do have sulfur-reducing bacteria. But then I had them in a file with some forested samples and a pedon from an urban area. And when I looked at the percentages, it really didn't have a higher percentage of those particular phospholipid fatty acids. It's just that overall they had so many more microbes and phospholipid fatty acids that just looking at how much, it made it look like, oh, this, maybe it has maybe there are a lot of sulfur producing, but as a percentage, it wasn't that different. Okay, so this is where we give all the different microbial groups in the units of picomoles per gram of soil, and this is probably the second or third most highly used piece of information. So just as looking as whether your soils are a measure of your soil health, generally think of the more Arbuscular mycorrhizal fungi, the healthier the soil will be, the more gram-negative bacteria, because it's there in the rooting zone, so you have more roots, you have more aggregates being formed. Eukaryotes and are the predators that are eating the fungi, or well, sometimes they eat fungi, the bacteria, and since they have lower carbon nitrogen ratios, the more you have of them. As they eat the bacteria, nitrogen is released, other nutrients are released. Fungi are also help with aggregate stability and performing other functions in soil. So usually the more the better of these also. You do have to remember that pretty much any of these except for mycorrhiza can also be pathogens. So if you have an excess of fungi, I have one client that will send in a set of samples, say good beans and poor beans. Occasionally the good sample will have a lot of fungi, and that they've figured out has been because it has a pathogen. That's not the normal type of samples that we get sent in, but I think you do have to know about that there are many roles for these different organisms in the soil. Okay, now another way of looking at it is the relative abundance. And so you may or may not find statistical differences with the values expressed as picomoles, but maybe they'll be significantly different when you're looking at the relative abundance. So sort of think of this as, what if we made a pie chart of these? So again, you usually think of the samples with higher AM fungi, gram-negative bacteria, eukaryotes, and fungi as being healthier. So I don't think there are any set values for what is healthy and what is not, because it depends on other soil variables, and I will discuss those a little bit later. So I would like to go back to this, the one where it's been in the picomoles per gram. So let's look at this data again. You notice that the fourth one has the highest level of AM fungi, and then the last one also has high levels. So a lot of this is appearing to be similar to what we found with the total mycorrhiza. But if you look at the fungi, this is kind of interesting. The third and fourth have a lot higher fungal quantities than the other samples, with the last one coming in at a third. And also the fifth one, pretty much across the board, has the lowest levels of all of them, of all the different types of microbes. So now let's look at where we have the relative abundance. So you see they've been much more equalized, and what's another thing that's kind of interesting, the sample, the fifth one that has the lowest amounts, actually, if you look at the mycorrhiza, the relative abundance is one of the highest, or the highest. So it may not have that many mycorrhizal cells overall, but because it has a lower number across the board, that as a relative abundance, it's fairly high in mycorrhiza. Even when you look at the relative abundance, the third and fourth samples still are high in fungi. The fourth and the last one, which so far have been looking the most healthiest, they have the most gram-negative bacteria, so they probably have a lot of rooting, and they are lower in actinomycetes also. And remember, the actinos are found more prevalent just everywhere. So that can be an indication that there are other types of organisms, not just the actinos. So another thing that I usually give the clients is a list of how MIDI is categorizing the microbes. So what this is a text file that just lists the different microbial types over on the left, and then for each one it lists all the different PLFAs that are categorized as that type of microbe, just so you know. Okay, so the next groupings we have is the fat types. Now, this looks like a lot more information, but you do need to consider that some of these fat types, or most of these are shown, are also microbial type indicators. Like the 10-methyls, those are the actinos. Something that I didn't bring up before, we found out several years into this that the extraction we're using doesn't allow us to look at aerobes, and those are indicated by the DMAs. So I have not used the color bars on those, so just ignore those values. But we do have other types of fats, like looking at the straight chains, the branched, the monounsaturated, the polyunsaturated. And again, the 18.2 is the same as the fungi. The 16.1 is the same as the arbuscular mycorrhiza. So the main new values that you have here, the 18.1 omega-9, and they've listed this separate because fungi have a lot of the 18.1 omega-9, and some people use that as an indicator, but it is not quite as reliable as the 18.2. So I do not classify that as a fungi. I have left it as general. I haven't put it into a specific category. What MIDI says they do is, if it is a forest sample, they put it in the fungi. If it is an agricultural sample, I think they put it in gram-negative. So I have left this out. If you want to, you can include it in. If you see reason why you have reason to believe that it would be another fungal indicator, you could add that in. The other one is, here we have the cyclos. Remember when we discussed the gram-negative bacteria? So here are the cyclos are given. So these are can be used, some people use this as sort of a stress factor, and we'll look at that a little bit later. So again, these are the same ones, looking at them as fat types. The new information would be the cyclose and the 18-1. So while it looks like a lot more information, a lot of it is really similar to the microbial types, because a lot of these do indicate microbial types. There are a few that are extra information. So again, I supply the text file that shows how all the different phospholipid fatty acids are categorized into these groups. Okay, so the final page are the ratios. So we have fungal to bacteria, predator to prey, gram-positive, gram-negative, saturated, unsaturated, mono to poly, and then the gram stress. So the gram stress, that's the precursors to the cyclose, those phospholipid fatty acids that have a double bond, that if they add an extra carbon or change to the cyclose, those are divided by the cyclose. So this is a relatively new piece of the software. We've just had it the last couple years. It's a different way of looking at the data. It may produce significant differences that you didn't see before. For instance, let's say you had a tendency to have higher fungal values in a certain treatment, and you tended to have lower bacterial values in that same treatment, but they didn't come out to be statistically significantly different. If you look at the ratio, you may find that when you combine those two factors, it may become statistically different. So in my opinion, the fungal, the bacterial, the predator-prey, saturated, unsaturated, and negative gram stress, the larger the values, the better, the more healthy the soil. The gram positive over gram negative, I generally think the smaller the better, because you have the gram negative on the denominator. So, so if you have more gram positive than gram negative, it's going to be above 1. If they're about the same, it's going to be around 1. So I consider the more gram negative, the better. There may be different reasons. Maybe you're looking at something different. And to tell you the truth, the mono versus poly, I am not sure. There are some indications that with more stress, you would have more double bonds to sort of shore up the cell membrane. But on the other hand, you have the polys are what indicate the eukaryotes, and the monos are the gram negative and the fungi. So I'm not really sure, but it would be interesting to see if you have differences. So just looking at the samples that we have, again those samples 3 and 4, they are higher in fungal to bacterial ratios. The number 4 has the highest gram negative stress factor. So here the higher is the better. You don't have as many of the cyclose. The gram positive to gram negative, the ones with the best ratios are the last one, and the number 4. So that's it for the data that we get. That's what we provide. The ratios, I also have a file that I send out that will show you how these were calculated and which values, like for fungal and bacteria, the saprophytic fungi, the 18-2, and the Mycorrhiza, the 16-1, are in the numerator, and then all the rest of these are in the denominator for the bacteria. So you can look at these to see how the ratios were figured. You could figure them a different way if you want to express ratios in a different way. Anyway, I provide you with how these ratios have been figured. So let's just quickly look at these samples. And I hadn't looked at what these samples actually were for a long time. I think I told you earlier that they're planted into warm season grasses as part of the CRP program. They were planted in 1990. Sericea lespedeza has been replacing some of the native grasses. It was farmed for decades before it was put into CRP, and then the CRP program expired for these particular fields. So the samples were taken out in 2018. So fields 1 and 2, that just shows different location. They were all pretty much on the same landform. Field 3, there's eroded and non-eroded. So if you'll remember, these are the ones that had the high fungal area. So they were really parts of the same field. They were different in that one part of it was eroded and one of them wasn't. And remember the number 5 sample, it was continuously cropped for 80 years. So that's why it showed the lower numbers of microbes. And then the last one was a degraded native prairie, which was invaded by shrubs, trees, and Lespedeza. And it's surprising to me that the south field, the uneroded one, was just as high or higher in a lot of these factors as the native prairie. A lot of times we will see that native prairie has twice the total PLFA of restored native grasses, even if it's been 20 years. I don't know if it's because of the invasion of the shrubs and trees and Lespedeza that it wasn't always showing higher soil health or higher microbial counts. But you know, not all data is perfect. So while this isn't perfect, I think you can see how you can use the data that you have and try to figure out. I think at this point, PLFA can inform you, and by selecting wisely what you're comparing, you can also learn how the PLFA is affected by choosing your treatments that you're going to look at. I also have some other data on the above here. The 2 fields with the most fungi have the lowest pH. I don't know if that's the only reason. I know sometimes when we have low pH, there usually ends up being more different before you see such a major difference in fungi. And if you look at the prairie, most of the chemical and physical analyses that we have up here, like total carbon, mineralizable nitrogen, active carbon, it's the highest across the board. What's kind of interesting is that the aggregate stability is actually lower than the 3rd and 4th samples. So maybe that's another indicator of the degradation, and maybe that's indicating too that there are lower microbe levels. Okay, so I believe I've gone over most of this. It's a 2-day extraction process. It takes 25 hours running on the gas chromatograph for us to get the actual results. We transform the data, download it, format it, and email it out to people. The software lists the individual PLFAs and gives the amount in picomoles per gram. We list the total PLFA. It's correlated with microbial biomass. They're classified by microbial types and gut types. Ratios are calculated, and these can be useful as well. Now, other things to know about PLFA. It was not developed as a soil health analysis. A lot of it was started about 30 years ago, more as a soil biology assessment. But I think there are a lot of reasons why you can use it. It does give us more information about the microbes, and through looking at it, we can learn about what is going on in the soil. There's people who spent their whole careers concentrating on PLFA, so there is a steep learning curve to it. I don't believe there's across the board good or poor values. There are some microbes that do not have PLFAs in their cell wall, so Archaea will not be included in this. I think at this point, comparisons and relative difference are what's valuable, so make comparisons. We don't usually suggest that farmers get this analysis because it is expensive and it's difficult to explain just a single PLFA sample without some sort of comparison or other background information. But for researchers, think how many data points you're getting with this analysis for the price. I think it's hard to find another analysis that's going to give you as much different information that you can use. Now, another reason why we don't suggest that we use it for farmers is because there are many factors that affect PLFA. The chemical properties, the pH, how much fertilizer, the other nutrient content, The more nutrients you have, a lot of times the less soil mycorrhiza you have, because the plants don't need it as much because the nutrients are available. Soil mineralogy, the parent material, the clay types, how old is the soil? Is it a prairie soil? Is it a forest soil? All of these things affect how many microbes you have. Physical properties like the soil texture, compaction, aeration, bulk density, the weather, and I should probably have climate too. Not only does the climate affect how many microbes you're going to have or what kinds you have, but just the seasonal weather or weather day to day. What's the temperature? How much soil moisture do you have? Will change the amounts of the microbes. Management, tillage will tear up the fungi, decrease those. The types of plant species will have different microbes living with them. Soil amendments, if you have added manure, that will increase the microbes. You're giving them more to eat. They vary through the time of year. So the microbes you have in the spring aren't going to be the same microbes that you have in the fall, which just kind of makes sense. But I had to actually see it on some of the data. It was grouped into, we have some statistical analyses that comes with the software, and I had it grouped in a dendrogram, and I had samples that were taken in the exact same plots, but by grouping in similarities, all of the spring ones were in one grouping and all of the fall ones were in another. So it mattered much more what time of year it was than what plot it actually came from. And as I've mentioned before, it changes, the microbial amounts and types vary as you go deeper into the soil. So we do use some quality control measures. We include a blank and a standard in every run to confirm that the batch was extracted properly. We vacuum dry or freeze dry the samples down to 5 microns for several reasons. One, so we have a consistent sample weight. Also, if you have more moisture in the soil, you probably affect how well it's extracted, because when you add the extractant, is it diluted by soil water or not? Another thing is we dry down the samples and then we freeze them, so you don't have the 3 degradations that can that will break down phospholipid fatty acids, which are light, heat, and moisture. So they've been dried down, they're in the freezer, and it's dark. So we can keep them for a relatively long time that way. We add an internal standard to the extractant, and it's a PLFA that's not found in nature, and it's a known amount. So that corrects for differences in the extraction process. Also, if we need to concentrate or dilute a sample, sometimes there's so much of a PLFA that it's beyond the detection level, so we have to dilute it. And because we have this internal standard in there, that can correct it back to what we know it was in the beginning. And this allows us to find out, it allows us to adjust and be able to run it at a concentration that we can measure it at. We convert mass to molarities in order to equalize the long and short chain PLFAs, because otherwise the longer chains are longer and heavier. So it would look like there were more of them because they would weigh more. But since we put it in a form of molarity, we're looking at numbers of molecules. We use a calibration standard, like you saw on the picture of the screen of the computer as we were running it, to calibrate the gas chromatograph as we're running it. We provide standard microbial group results as an output. What you can do is, because we have all of the PLFAs listed, you can classify the PLFAs in a way that you want to. You don't have to use that we did. We work closely with the Median Corporation to keep up to date, and then we contact each other back and forth and ask each other questions. There are some limitations to PLFA. It only gives an indication of who the microbes are, the general groups that you've seen. We don't identify the species. We don't really know what the microbes are doing in the soil, and since we don't know the activity, we don't know what level or how active they're being. We just know that they are there. So you may want to look at some other measurements of activity in addition to this analysis. We do provide relative biomass measures. What we give you is just how many picomoles of the phospholipid fatty acids that indicate for the groups, or the fats, just what is in there. That doesn't tell you how many grams per gram of soil of fungi there are. There are conversion factors that you can use, and I think that would be instructive, because if you remember, little tiny gram-negative bacteria have 2 layers of phospholipid fatty acid bilayers, whereas if you have fungal hyphae, they probably have a lot fewer phospholipids per total body weight than a gram-negative bacteria does. And while it might be instructive to look at these, I wouldn't use this for statistical analysis, because I'm sure each species probably has a different ratio of biomass per PLFA picomoles. And what you would be doing would be introducing area. I hope you understand that. If you need me to, I'll try to explain that a little better. There are some references that are helpful. This is on the left. We have high throughput phospholipid fatty acid analysis. This is the extraction method that we used. We went to Jeffrey Byer's lab and he showed us how to do it. Then there's another useful article, The Use and Misuse of PLFA Measurements. It gives you the pros and cons of using different measurements, what are the most reliable, which ones you should take with a grain of salt. Also, both of these will have other listings of articles that you could go, the references they use, so you can look up those references as well and learn more. There's a Wikipedia article, the phospholipid-derived fatty acids. That's very helpful to look at. I also usually email out initial exploratory PLFA data analysis. This is not published, but it's something that our coworker, Kristen Wiem, who works for the ARS, put together for us. She went with us to be trained at the ARS lab and MIDI's lab. So this is also very helpful.